Hello and welcome. Have you ever wondered how scientists measure the amount of a substance in a liquid, even when it's completely invisible? Today, we're going to explore a foundational technique in biochemistry that does exactly that: photometry. In the simplest terms, photometry is all about measuring light. Specifically, it's a technique used to determine the concentration of a substance by measuring how much light it absorbs. Think about shining a flashlight through a glass of juice. The darker and more concentrated the juice, the less light will pass through to the other side. Photometry is the scientific version of this simple observation. The primary instrument used is called a spectrophotometer. This machine has a light source, a component that selects a very specific color or wavelength of light, a special transparent tube called a cuvette to hold our liquid sample, and a detector on the other side to measure the light that successfully passes through. The entire process is governed by a principle known as the Beer-Lambert Law. While the name sounds complex, the idea is straightforward: the amount of light a substance absorbs is directly related to its concentration. More substance means more light gets absorbed. The spectrophotometer gives us a reading called absorbance, and a higher absorbance value means there's a higher concentration of our target substance in the cuvette. This simple principle has massive applications in biochemistry. For instance, one of the most common uses is to measure protein concentration. Scientists can add a special dye to a sample that turns blue in the presence of protein. The deeper the blue color, the more protein there is. The spectrophotometer precisely measures the intensity of that blue color, allowing for accurate protein quantification. Another key application is in studying enzyme kinetics, which is the speed of enzyme reactions. If an enzyme creates a colored product, we can put the reaction mix in the spectrophotometer and watch the absorbance increase over time. This tells us exactly how fast the enzyme is working. Finally, photometry is crucial for working with DNA and RNA. These molecules naturally absorb ultraviolet, UV, light at a specific wavelength. By placing a DNA sample in a UV spectrophotometer, we can instantly measure its concentration and even assess its purity, a vital step for experiments in genetics and molecular biology. So, from proteins to DNA to enzyme speeds, photometry is the workhorse of the biochemistry lab, allowing us to see the unseen by simply measuring light. Thank you so much for watching this introduction to photometry. If you found this video helpful and want to learn more about the amazing tools of science, please consider liking this video and subscribing to support the channel. Your support helps us create more content just like this. We'll see you in the next one.